Hi everyone, my name's Joe and I did my data science final project on some neuroscience data, more specifically on RNA sequencing of different cell types in the brain. To introduce that topic, I have to introduce this website called brainmap.org, and essentially it's just a repository of thousands of different experiments done on human and mouse brains, and all of the data acquired from these experiments is available for download. So as you can see here, CSVs and zips, among others, are downloadable, and these two pieces of data are actually what I based my project on. And before I get into that, I want to talk about a different data set that I downloaded before this that didn't go too well. So as you can see here, there's some missing information, and the way I sought to deal with this missing information is I thought, well, maybe I could take all of the records that have missing fields and just omit them. And so, as you can see, what was already a small number of records became an even smaller number of records. And then I ran some preliminary predictive models, and they all looked pretty bad. So I just ditched the dataset altogether. So here you see a different dataset. It's full, there are no missing fields, and it's also significantly larger. So this is what I based the project on. And the last column of that dataset is an extension of a link, and it's based on that brainmap.org. So if you follow that link manually, it'll download a file, and if you read that file into R, you'll see that there's a whole bunch more information, a lot of information, for each one of these files. So 15,000 of these files, there's one of these. So I thought maybe I could read each one of these in, do some calculations, and spit them back out as values that I can add as columns to the original data frame. And so that's what you see here. Here's a list of all the URLs, and if you concatenate brainmap.org to that extension and read it in as a data frame, you can calculate some things. These are just efficiency measures, and you can output those efficiency measures. So now you have this length and density, which become two more columns, which you can add as columns to the original data frame. Now, I didn't actually run this because, well, I tried, but for 15,000 files and 45,000 rows per file, my R just couldn't handle it. It would just keep crashing. But I ran it for two of them. As you can see here, you get a length and a density, and theoretically this would just run down 15,000 times, and you would add these two columns onto the original data frame. So the next thing I sought to do was do some exploratory analytics. So I tried to run pairs.panels, but it wouldn't render. So instead, I individually plotted some histograms and did transformations that way. So this is before and this is after transformations for some of the variables, and this is what the distributions looked like afterwards. So let's go ahead and run that real quick. So, yep, here you can see the distributions. And so the next thing I wanted to do was deal with outliers. So I called an outlier anything that was 3 or more standard deviations away from the mean. And so I ran that function that calculated that over the entire data frame, and it told me which ones were outliers or not. So as you can see, some of these are outliers. So what I did was I took any record that had an instance of an outlier, and I just deleted it from the original data frame. So as you can see here, I mutated the original one to have all but the rows with outliers. So let's go ahead and run that. And so here you see the distributions after the outliers are removed. And you can see that a little bit better here. Now, next thing I wanted to deal with was collinearity. So I did pairs.panels of all the numeric data now. And as you can see, there are some very strong correlations between some of the variables. There's this concept called variance inflation factors, and essentially they can be used to measure collinearity. And so if you come over here and we run this VIF on a random model, we get these numbers, and these numbers are those VIFs, and a large number corresponds to a high chance of collinearity. So as you can see, this number is pretty large, this number is large, and this number is very large, at least relative to these. And so what I thought I could do is maybe I could remove this one and this one or this huge one and get rid of that multicollinearity. So that's what you see here. When you run it without those two, you get rid of all of it. So all these numbers are small. And then same thing here, you lose it. So this is just something that I wanted to keep in mind when I created my models later, to keep in mind the potential multiple collinearity. So next thing I wanted to do was store it in a database. So I took all of the columns, because they all have important information, and I put them into a table in SQLite. But I don't need all the columns for the analysis portion, so when I retrieved it, I only retrieved the columns that I was going to need. So I think I have to remove them first. And now we can run it. So here it is. You can see I pulled out just the ones that I need. So now we're going to stay in R for the rest of the presentation. So the next thing I did was created training and testing data sets from the original data set. So I used sample to randomly pull out half of the indices and run that across training and testing. So when you run that, this is the number of rows of each one, so you see that they're essentially the same. And so now we have training and testing data sets. And so the first model I wanted to create was a linear regression. And specifically what I was interested in is one of the columns in the data set has this not aligned RNA sequence. And so normally you want to align your RNA sequence with either another part of the genome or another genome. So it was interesting to me that this column was not aligned to anything. So I wanted to see if that correlated with anything. And when I ran this model, I got this R square that was huge. It was so close to 1, so it had strong predictive power. And if you run it, you can see there are tons of measures of this statistical significance down here. But as we can recall from the VIFs, there was multicollinearity, so we needed to deal with that. So the same way I dealt with it up there, I took out here either one or another of those factors that was likely leading to multicollinearity. So we run these two models, and you look at their R squared values. You see that model A has an R squared now of 0.9081. Didn't go down that much, but this second one has an R squared of 0.7. So we're going to roll with model A. So the next thing I wanted to do with model A was backfit it. So essentially I took all the values that weren't statistically significant, all the variables, and just get rid of them. And the next thing I wanted to do, well, I guess this is all of them. But in the end, the adjusted R squared doesn't go down very much. It's still above 0.9. But the last thing I wanted to do was tune this model. So now I only have variables that are statistically significant, but of those variables, there are categories that might not be. So here you see donor age. Some of them are statistically significant, but not all of them. So we're here to tune the rest of the model. I just got rid of everything that wasn't statistically significant. So in the end, we have two models. We have a model where we backfitted and a model where we cut down to just whatever was statistically significant, and its R squared is still above 0.9. So now we could evaluate these two models, model A, which is the backfitted one, and the tuned one, which has everything that isn't statistically significant taken out, and we can compare them. So here we have an anti-log function, and this is because we transformed the data early on, so this is to transform it back. So we're going to take the testing data set and all the not aligned values and bring them back to the original form. And so here we make some predictions based on model A and based on the tuned model. And so when you calculate the MADs of these two models, you get these two values, this one and this one. I guess they should be down here. Yeah, so they're very close to each other. So these models are very similar. And when you calculate the mean squared error, you also get very similar values. And so the tuned model might have a slightly larger MSE, but the difference is so small that they're essentially the same. And since the tuned model has fewer variables that it uses to predict, it's the preferred one. So the next model I created was a logistic regression model. Here, I recalled that, let's see, if we open the original one, the original data, you can see here that there's this VIS. So VIS indicates a particular brain region, and if you go far down, there should be an instance of ALM. They're the two brain regions that I took these data from. So I thought, well, maybe since I took from two distinct data sets and I put them together, maybe there is a difference between these two. So what I did was I took from the original data set, that column, and I created a new column and added that on, indicating which ones were VIS and which ones were ALM. And then that's going to be our response variable in the logistic regression model. So there that is. So now we mirror that onto the training and the testing data sets. So here we have the logistic regression model, the first one, and we have all this statistical significance, interesting things. And so the first thing we did was backfit it. Oh, where did that go? So here, we took out all the things that were statistically significant, all the variables. And now we have our last model, which is this. So same idea, now we have the donor age, where some of them are statistically significant, but not all of them. So our tuned model, which is down here, is essentially just taking out everything that doesn't have an asterisk. So there that is. And now we're going to do a similar thing where we compare the two, except for logistic regression, we're going to look at their accuracy when they predict on the testing data set. So here's the logistic model prediction and the tuned model prediction. And so we're going to take all the instances where there's it was calculated more than 50% chance that the value was VIS. And we're going to apply them onto here to calculate whether it was VIS or ALM. And now we're going to map it back to the original to see how many of those were correct based on the testing data set. And as you can see here, they both tested better than chance. They were better than chance at predicting these values, but they weren't great. So maybe there's something different that I could have done by either changing the bias when I was creating the testing versus training data sets, or etc. So, well, I guess that's my project. Thank you guys for listening.